Okay, hello Bio 348 students. This is Candice, your TA. I'm just gonna do photosynthesis for you in this format. I'm gonna go through the slides of a voiceover. I'll make sure that these are up on YouTube as well as Moodle. They are gonna be very long, so I don't expect you to just sit and listen to my voice for like hours and hours. I don't even like hearing that. So take your time. You will have this forever until the end of the module to go through, to sit, to listen to it. You can listen to it as many times as you want. But this is really just a revision section. A big chunk of it is revision, and then the last little bit is building onto your already known knowledge. So we're going to set the foundation, or the foundation should have been set in Bio 204. And what we're going to do is we're going to revisit it, add a little bit extra, make sure we understand everything, and then we are ultimately going to add at the end onto it to ensure we understand photosynthesis, especially in terms of applied ecophysiology and taking into account physiological and ecological considerations, as well as potential abiotic situations. So just be warned, a lot of this will sound like revision, but if your foundation's not solid, the rest of this module is going to be wonky. So let's get started. Now, remember, these are from Taser and Zager, the book, your prescribed textbook. We're looking at chapters 7 and 9. And if a graph or figure isn't fuzzy or whatever, it probably is in Taser and Zager. So do yourself a favor, have a look at that textbook. And work through chapter 7 and 9 with this, my lovely voice in the back of your head, in your ears, like an ear. Okay, so this is all revision. This is building up from Bio 204. So we're going to recap and then we're going to add. First thing, obviously, photosynthesis, energy derived from the sun. Phototropism, remember phototropism, photo being the photons and tropism to move towards. So for plants, they need light. Light gives them their energy in order to survive. Okay, photosynthesis is the only biological process that can harvest energy from the sun, and through photosynthesis convert it to chemical energy. So from light energy to chemical energy, which they use in their daily run-of-the-mill of their or processes that they need to go through. Remember, at the end of photosynthesis, we have carbohydrates, and that carbohydrates fuels. So you should know this equation. This equation should not be unfamiliar to you. You should know this equation back and front because photosynthesis is in this direction, whereas cellular respiration is in that direction. So we have glucose plus oxygen and we get carbon dioxide. So cellular respiration this way, photosynthesis this way. Now please remember that plants also respire. So plants photosynthesize, they go this way, and plants respire. So they give off carbon dioxide. But the photosynthesis that they do, it's almost like a business where when you have a business and you're organizing your profit margins, you will take the cost of your business and you'll minus your cost from what you earn. So you would have marked up everything and you would get a net. So the photosynthesis pays for the cellular respiration, for the carbon dioxide released by photosynthesizing more than what is needed. So we have a profit of photosynthesis taking place as opposed to giving carbon dioxide. So just bear in mind, plants will photosynthesize and they will cellular respire as well. However, the cellular respiration effects are mitigated and squashed by the photosynthesis. And remember that this basic transformation into glucose establishes the energy sources for all life forms going forward in the food web. If you're a vegetarian, direct. Vegan, direct. If you're a carnivore, then obviously indirect. Okay, remember that the leaves are responsible for photosynthesis. That photosynthetic tissue is the most active, and the cells that are most active or tissue is this mesophyll tissue. Remember, what makes the plants green is this chloroplasts, and the chloroplasts have green absorbing pigments called chlorophyll. That's not the only thing in the chloroplasts. There's different types of pigments as well, which you should remember, that will aid in the photosynthesis. Our end result of photosynthesis is primarily sugars. Remember, this takes place in the thylakoid. In the chloroplasts, we have thylakoids or granum that are broken up, or granum is a complete package of thylakoids, and it's in these thylakoids, on the thylakoid membrane, that we're going to have a light-dependent reaction, and it's in the stroma where we are going to have the Kelvin-Benson cycle. Remember, photosynthesis is split up into two parts. The light-dependent reaction, light-dependent meaning it needs light in order to function, and the dark or the Kelvin-Benson cycle. The light-dependent will take place in the thylakoid membrane and the Kelvin-Benson in the stroma. Okay, now the reason why the light reaction needs to happen in the thylakoids is it needs a semi-permeable membrane to allow for chemoosmosis. So we'll get to it, but the reason why it's on the thylakoid membrane is for chemoosmosis. So this is the structure of a leaf cell, or of a leaf, sorry, and we need to know it in order to understand the terminologies that are going to come. You start off with a cuticle, and this mesophyll photosynthetic tissue, the palisade, spongy layer, etc. This is where the photosynthesis is going to take place. Your chloroplasts. Do not get confused. A stoma is another word for stomata, and that is different from the stroma, which we find in the chloroplasts, okay? And remember that our guard cells are what is going to allow the stomata to open and close, and that will be done via water. So it will take up water or release water content to open and close the stomata. Remember, on Monday, well, whenever you're reading this, our first practical, we had a look at C3 and C4. Now, the structure of this leaf, or of leaves, differ between C3 and C4 plants, and it is the way they are structured that allows them to do what they need to do. It is the structure of these leaves that will make the photosynthetic pathways unique, and you are going to have to, in that first practical, discuss the differences between these two in order to explain why the C3 would do better than the C4, or either way, either or, whichever one your data shows. So remember this palisade mesophyll, the spongy mesophyll, that is our mesophyll tissue, that's our photosynthetic tissue, and you can see how in the C4 this vascular bundle changes, and the way we have the bundle sheath and this bundle sheath over here, and how the palisade mesophyll is around it. And this is going to be very important because the light reaction is going to take place in the mesophyll, and the Calvin-Benson cycle is going to take place in the bundle sheath. And it's the separation of rubisco from where we will have access to a carbon dioxide. So carbon dioxide comes in, and it's all, and in the C3 the carbon dioxide has access, or the light reaction and the Calvin-Benson cycle is exposed to this carbon dioxide, whereas here the carbon dioxide comes in, it's only exposed to the light reaction. The light reaction converts everything into malate, and the malate transports it to the bundle sheaths, and that's where the Calvin-Benson cycle is going to take place. And if all of this is sounding unfamiliar to you and you're going, what the hell, I don't remember this from Bio 204, don't worry, we're going to go through it, we're going to make sure you understand. But what you need to know from the outset is that the way the plant is structured, the way the leaves are structured, is really going to tell you the difference between C3 and C4. And the reason for this is to protect the rubisco enzyme, to keep it away from oxygen, which we'll get to. So here is a picture of a chloroplast. We have our cells. In our cells, we have our chloroplasts. There's a picture of a chloroplast. Here you can see our granum, which is a stack of thylakoids, individual thylakoid. Going to come, if you see over here, we have the thylakoid, and the thylakoid membrane is where the light reaction is going to take place. And in here we have the stroma, and the stroma is where the Calvin-Benson cycle is going to take place. So this slide is to show you the location, where's things happening. Now remember, photosynthesis is broken up into two sections, the light-driven reactions, or the light reactions, and the Calvin-Benson cycle. Just because this is, please don't get confused, this doesn't happen in isolation of each other. So one doesn't happen during the day and the other one at night. So sometimes in some textbooks and some YouTube videos, they might call this Calvin-Benson cycle the dark reaction. It's not. It happens simultaneously. The only place where that would be significant or different would be CAM photosynthetic pathways, which we will get to later. But so just bear in mind that we say light reaction because there needs to be light in order for this process to occur. And I'm calling this the Calvin-Benson cycle. Other people might call it the dark reaction. That doesn't mean it happens at night. It happens simultaneously, except in CAM. Okay. So remember the basics of photosynthesis: water, light, gonna come in, and in the light reaction, that light is gonna break the water molecules, and we're gonna take the electrons from hydrogen. We're gonna be left with 2 positive ions, and we're gonna be left with oxygen. The oxygen, remember, it's unstable. It doesn't like being alone, so it's going to attach to an oxygen, and we're going to get our oxygen 2, which is going to be released. So remember from Bio 204, the oxygen does not come from the carbon dioxide, it comes from the water. So the carbon dioxide is not used in the light reaction, and the light reaction is what gives us our oxygen that we breathe. So remember, that oxygen that we breathe comes from water. The process of photolysis, which is the splitting of the water molecules, is where we get our oxygen from. We're gonna get 2 hydrogen plus ions that are gonna be floating around in the thylakoid membrane. Here they are over here. And then we're gonna go down. The electrons are gonna get given to photosystem 2. They're gonna be excited by light. It's gonna give it that energy. It's gonna cascade down the electron transport chain. As it does that, it's going to release energy, hence pumping the hydrogen positive ions through that thylakoid membrane, that chemoosmosis, and then it's going to go to photosystem 1. It's going to get a revitalized energy, and then depending on which cycle it's going to take, whether it's going to be non-cyclic or cyclic, it will either go back to the electron transport chain and cascade down again to pump more hydrogen positive ions, or it's going to be given to NADP. And that NADP is going to be converted to NADPH, which is going to go off to the Calvin-Benson cycle, along with the ATP synthase protein pumping from NADP to ATP. So our byproducts for the light reaction are oxygen, which gets released back into the atmosphere, NADPH, and ATP. That NADPH and that ATP are sent off to the stroma. In the stroma, we have the Calvin-Benson cycle. The Calvin-Benson cycle goes through 3 stages: carboxylation, reduction, and regeneration. And from that, we're going to go along. So the NADPH, the ATP, plus that carbon dioxide now that we breathe out, it's going to come into the Calvin-Benson cycle, and we are going to get a glucose molecule at the end of it. All right, simple, easy peasy. Let's go. Every time we're going to add a little bit more. We're going to add a little bit more until you know this 100%. Okay, so everything I've just said in slide form now. The light reaction, remember we have 2 photosystems. We have photosystem 2 and photosystem 1. Don't get confused. They were named in the order that they were found. So photosystem 1 was discovered first and then photosystem 2, but in actual fact, in the light reaction, photosystem 2 comes first. So it's first photosystem 2, then photosystem 1. And so we have these 2 photosystems that convert light energy into chemical energy. Remember that we create that energy, or that energy is used, or the power that transfers that energy is through the electrons that were snatched away from hydrogen and we're left with the hydrogen positive ions, but the photosystems take that electron, and it is through that electron that we get the energy that is required. And remember, majority of the electrons is used to reduce NADP. Remember, when you reduce, look at that D. When you reduce, you're adding. When you oxidize, exiting. So when you reduce, we're adding that energy from that electron to combine to give us NADPH. So that energy from there to reduce NADPH to plus to NADPH, NADP plus to NADPH, and oxidize, where we take away the electrons and the hydrogen ions to leave us with an oxygen. It binds with another oxygen and we get our oxygen 2, which is then released back into the atmosphere. This YouTube video is very helpful. The guy is a little bit annoying, but yeah, he's alright. So if what I'm saying is not gelling with you, try this YouTube video and hopefully it gives you a little bit more understanding. So let's look at the light reaction. Remember, here's our chloroplast, there's the granum, there's the stroma around it, there's our thylakoid, and we're zooming in, there's our thylakoid membrane. This is the inside of the thylakoid membrane, and this out here is the stroma. Here's our photosystem 1, our photosystem 2, and our photosystem 1. Here's our electron transport chain. And what's going to happen is we're going to have the light coming in. We're going to have photolysis here. Photolysis, or an oxidization of water, where we're going to have the oxygen forming to give us oxygen 2, and we're going to get 2 hydrogen ions, and we're going to have the 2 electrons from those hydrogen ions going into photosystem 2. Now, photosystem 2 is filled with chlorophyll, chlorophyll A, chlorophyll B, carotenoids. All of those pigments are in there. And specifically at that light reaction center, that P680, that light reaction center, it's going to absorb the light. Now remember, we'll get to it, but 680 nanometers is more energy than 700, so just bear that in mind. So photosystem 2 can be written as PS680, and photosystem 1 can also be written as PS700. So that is just the wavelengths at which they absorb energy. So photosystem 2 absorbs energy at 680, and photosystem 1 absorbs energy at 700 nanometers. Remember, there's a reaction center in there, and what is happening is the light hits the electrons in there, the electrons sitting at the reaction center, which is here by 680, and the energy is passed from one chlorophyll pigment to the next chlorophyll pigment. It's excited, it's gearing up, it's got energy, it's filled with energy. It gives it to this reaction center, and the reaction center gives it to the electron, and the electron jumps up. Then here comes plastioquinine. You should remember all this. Plastioquinine comes and collects the electron that's excited, that's full of energy, and as it collects it, it releases some energy. And it collects it and it gives it to cytochrome B6F. And again, cytochrome B6F gives it off to plastocyanine. And as it is cascading down this electron transport chain from protein to protein, it's releasing a bit of energy. And as it's releasing a bit of energy, the hydrogen ions are going to pump through the thylakoid membrane. And as it's pumping through, they're going to change. They're going to cause this energy, and it's going to pump the hydrogen ions back in, so that when we get to ATP synthase, it can be pumped out to give us our ATP. But we need this energy that is being released by the electron transport chain to pump this ATP synthase, because remember, we're going to go from a high concentration to a low concentration. So all those hydrogen ions that are coming back from Calvin cycle, we need to make sure that we have a big, strong hydrogen concentration over here. And this energy that is released is going to allow the hydrogen plus ions to pump through. Once we get to plastocyanine, it's going to give it to photosystem 1. We've lost quite a bit of energy, so this photosystem 1 also has chlorophyll A, chlorophyll B, has all sorts of pigments that are used for light harvesting, and it harvests, it picks up that light, it revitalizes that electron, and then all of a sudden, from photosystem 1, we have ferredoxin, and ferredoxin has got a choice. Ferredoxin is either going to give it so that it can change NADPA or NADP plus to NADPH, or it's going to give it back to cytochrome B6F. This will be the cyclic phosphorylation, and this linear movement will be non-cyclic. So if it gives it to NADP plus, which transports or makes it into NADPH, it's non-cyclic. If the ferredoxin decides to give it back to the cytochrome B6 so that it can cascade back to allow those hydrogen positive ions to come in, and also to give a little bit of energy to this ATP synthase in order for us to get ATP. So without the energy from the cyclic phosphorylation, we won't have ATP. So those are the two pathways it can take. This should not be unfamiliar to you. You should be remembering this stuff. Then we've got NADP being transformed into ATP. This ATP, the NADPH, are going to go off to to the stroma. They're in the stroma. They're going to go off to the Calvin-Benson cycle for carbon dioxide to be fixed. And the oxygen is going to be released out of the stroma, out of the chloroplasts, out of the leaf, into our atmosphere. So the byproducts of the light reaction is our oxygen we breathe, ATP, and NADPH. NADPH, ATP are going to go off to the Calvin-Benson cycle. And once they're done in the Calvin-Benson cycle, and the Calvin-Benson cycles use that energy, NADP plus is going to come back, and NADP is going to come back to get energy again from the light reaction. So the job of the light reaction is to give the energy that is needed to assimilate carbon dioxide to the Calvin-Benson cycle. So light reaction's job is to give energy to the Calvin-Benson cycle. Calvin-Benson cycle's job is to assimilate or to fix the carbon dioxide so that we get that delicious sugary glucose at the end. Okay, what I've just said in slide form. So we've got the Calvin-Benson cycle. Now, this concept of photorespiration, you should all remember Rubisco. Rubisco. Rubisco is that enzyme in the Calvin-Benson cycle that binds with carbon dioxide that allows us to fix the carbon in the that allows the plant to fix the carbon. So Rubisco is that enzyme. Remember, enzymes work on a lock and key, and Rubisco is that enzyme that connects with carbon. So Rubisco is a 5-carbon molecule, and the carbon dioxide that comes is 1 carbon. So we get 6 carbons. It breaks up which we'll get to later. But the point I need you to understand right now is that Rubisco is an enzyme, and Rubisco, you can see that not a very nice boyfriend. He's holding carbon dioxide's hand, or holding carbon dioxide's hand, but he's looking and checking out oxygen. Rubisco has an affinity for both oxygen and carbon dioxide. So what I mean when I say that is that Rubisco can bind with carbon dioxide, and it can also bind with oxygen. Now remember, it works on a lock and key. So if oxygen binds with Rubisco, that means that Rubisco molecule is finished. It's finito. It's not going to work. It's broken. In order to work, it needs to combine with that carbon dioxide, and it needs to go through the Calvin-Benson cycle, and process needs to occur. So when we go back to C3 and C4, C3 plants were really designed or evolved under high carbon dioxide levels. So we've got high carbon dioxide levels, it evolved, the Rubisco is working fine, and now all of a sudden we have oxygen. And the oxygen is starting to influence and flirt and bind with that Rubisco, making that Rubisco unusable, which puts the C3 plants at such a big disadvantage because the Rubisco is given access to both the oxygen that is released in the light reaction as well as the carbon dioxide that is needed for the Calvin-Benson cycle. So C4 plants evolved. So once the oxygen started filling the atmosphere, we started getting this problem. C3 plants started suffering. We come and they evolved into C4 plants. C4 plants separated Rubisco because Rubisco could not be trusted with oxygen. Couldn't leave Rubisco alone. Carbon dioxide couldn't leave Rubisco alone with oxygen. They were untrustworthy together. So the C4 plants have now made the light reaction occur in the mesophyll, where the oxygen is formed and then the oxygen is released. Malate is produced and malate is produced and it is pumped into the bundle sheaths, and it is in the bundle sheaths where the Calvin-Benson cycle will take place, therefore keeping the carbon dioxide and the oxygen separate and keeping the Rubisco away from the oxygen. And that's what makes C4 plants able to handle high oxygen levels. So now think about it. Think about it long and hard now. You've got an extra step in CO4. You've got an extra step of that malate, which is that taxi, which we'll get to, which transports to the bundle sheaths. If I increase carbon dioxide, which one's going to perform better? Which one's going to perform better under higher carbon dioxide levels? We've got an extra step here, and we don't have an extra step. More energy needed here, less energy here. Think about it. We'll answer the question later down the line. Okay, so when I say photorespiration, I'm really talking about that I'm talking about that Rubisco. I'm talking about that Rubisco binding with the oxygen and becoming unusable. In order to avoid that, we had C4 and CAM photosynthetic pathways coming about. And what these two do is they store and they transport into different units. The CAM plant will store, and the C4 plant will transport. But we'll get to that a little bit later. Now, we've discussed the light reaction. We're coming into the Kelvin-Benson cycle. Remember, the Kelvin-Benson cycle's job is to assimilate. Please don't get confused. Again, this is a term that means the same thing. If I say assimilate, if I say fixate, it's the same thing. The Kelvin-Benson cycle's job is to assimilate or to fixate the carbon dioxide to get it from that high value, that high oxidation state, to the lower levels, or to decrease the carbon dioxide from its highest to the sugar. Okay, so to get that carbon dioxide to assimilate so that we can get ultimately our glucose molecule. Not that simple, which we're going to get to. So Kelvin-Benson cycle has 3 stages. Remember, these stages will occur in the stroma. These are the 3 stages: carboxylation, reduction, and regeneration. Now, you need to know the numbers. You should know the numbers from Bio 204. But ultimately, carboxylation, this is where we have our ribisco. Here's our ribisco, ribulose 1,5-bisphosphate, or ribisco, or RUBP. And this ribisco, this is our enzyme, and we have the input, which is our carbon dioxide. It's coming, it's connecting with the ribisco. Remember, the ribisco is a 5-carbon molecule. This carbon dioxide is a 1-carbon molecule. Together they form a 6-carbon molecule. We're then going to get reduction, where our energy, that ATP and that NADPH, is going to be used to break up our 6-carbon molecule into 3-PGA or 3-PA, and then to further reduce it, reduce it from our 3-PGAs to 2G3P. It is going to be this G3P, which is going to be transformed and reduced further into our sucrose down at the bottom. This 2 G3Ps need to then be used. The AT is used to regenerate to give us our 5-molecule ribisco back. But the numbers don't add up. The numbers don't add up. Okay? Because we need a 6-carbon molecule, because glucose is a 6-carbon molecule, right? Okay, because it's a 6-carbon molecule, the numbers here don't add up. 5 plus 1 is 6, but I still need to make 5 again, so I've got to get 1 off. So what happens is this Kelvin-Benson cycle happens simultaneously with others in a factory, 6 at a time. Okay, so we're going to get 1 molecule coming, 1 carbon getting formed here, getting chucked with another one from another Kelvin-Benson cycle, and another Kelvin-Benson cycle, and another one, and another one, and another one, giving us our 6-carbon molecule, which is glucose. And in that same way, so we need the Kelvin-Benson cycle to essentially rotate 6 times before we get 1 glucose molecule. So it happens at the same time. 6 Kelvin-Benson cycles happen at the same time, and they work together to get those molecules, that glucose molecule, off, and each one will then be able to use ATP to regenerate it into our 5-carbon molecule. That is very confusing. I know it is confusing, believe me, this YouTube video is brilliant. It's a world's smallest factory, and it shows you step by step what I'm trying to say here. It shows you how these six Calvin cycles work in unison with one another, so that at the end of it, we end up getting a 6-carbon molecule, and we still end up getting the rubiscos being regenerated at the top. Please watch it, understand it, so that you understand the numbers. This Calvin-Benson cycle is broken up into 3 parts: carboxylation, reduction, and regeneration. It has to work in conjunction with others in order to get our glucose molecule at the end, so that we can end off with that ribulose 1,5-bisphosphate. Now, the whole purpose of this cycle, when I say cycle, is that it regenerates itself. If that rubisco is not formed again or regenerated back from G3P, it's not going to work. It's not sustainable. And this video shows you brilliantly what I mean when I say sustainable. I just don't have the elegance that this YouTube video is. Just very simple, and it explains those numbers to you. So watch it, please. You need to understand completely that what's going to happen is, because remember I'm saying 6 work in conjunction, so we're actually getting 6 times 5 is 30. 30 plus 6 is 36. 36 breaks up at PGAs, and etc. etc. Now, it takes 2 ATPs and 2 NADPs, H's, to do this phase, which means in order to get a glucose molecule, we need 12 ATPs and NADPH, and then we need a further 6 ATPs to regenerate back into rubisco. If this is confusing, watch this YouTube video. It'll explain the numbers, but you need to really understand that this Calvin-Benson cycle doesn't just happen on one turn. You need to understand the numbers. It's a numbers game. You need to understand it. You need to be able to realize. Okay. So now we are moving away from revision. We're going to start putting pieces together. We know the light reaction now. We know the Calvin-Benson cycle. We know the nitty-gritties. So we're going to start considering what's going to happen when we add other elements. Okay. So remember, recap, we've got a conversion of solar energy to chemical energy to get our glucose molecule, and it needs an electron transport chain. These processes are continuously occurring, and remember, it's working together with other Calvin-Benson cycles to go. The impact of the environment on this photosynthesis is so important, because if photosynthesis stops working, the plant, not only will the plant die, but our future energy sources are compromised. So when it comes to understanding how the environment, or how abiotic stresses, biotic stresses, how the climate change is going to affect plants, we really need to focus on photosynthesis, because if tongue twisters, if photosynthesis breaks down, the plant will die. And if photosynthesis, oh my word, if photosynthesis stops working, ultimately, no food, no energy, we're doomed. So it's important, and remember, Photosynthesis is where we're going to get our yield from. A yield being the fruit, the crops that are produced, how much, whatever. You want a better yield? Make photosynthesis more efficient. You want yummier fruit, you want bigger fruit, you got to make photosynthesis more efficient. So we have to understand how photosynthesis and the environment work together in order to uh, give us what we need in order for plants to survive. So, things we need to consider. We need to consider light because without light, no light reaction, no energy. We need carbon dioxide because with no carbon dioxide, no glucose. Temperature is going to be affected because remember, what is rubisco? Rubisco is an enzyme. Enzymes become inactive at lower temperatures and denature at higher. The same thing with proteins. Remember the electron transport chain? It has proteins, plastoquinine, cytochrome B6F, photosystem 1 and 2. They're all proteins, and proteins will be damaged under temperatures. Remember, humidity, soil moisture that controls the stomata, which allows the carbon dioxide and the oxygen to flow in and out, becomes a big problem in seed plants if it is closed and we have high internal oxygen levels because the oxygen levels are going to start flirting with the rubisco. And then other things that will influence photosynthesis, obviously abiotic stresses, which we'll deal with later. Remember, we've got the main process of photosynthesis. Remember, the light reaction is just to produce the energy that is required for the Calvin-Benson cycle, and this is the metabolic properties that are important in photosynthesis in the Calvin-Benson cycle. If this breaks down, we're having problems. The plant's having problems. That is at rubisco. Remember that affinity, it can either form with carbon dioxide and work, or it can bond with oxygen and just stop working. And oxygen, a home wrecker, and it just wrecks everything. Also, if we do not get this regeneration of ribulose 1,5-bisphosphate, no more Calvin-Benson cycle. If we do not get the metabolism of those 3 PGAs into the G3Ps, if this metabolism doesn't occur, we're not going to get the glucose that we need. So, tragic story I feel like sharing with you. In my second year of university, I spoke to a lecturer, I won't tell you who they were, at EMVS, and they said to me, no, do economics. It's a good first year module. It's easy. Do it as an elective in your second year. And I said, oh, okay, it'll be easy, it'll be fine. And there I was, nearly failed, crying. It was torture. And what I learned there, never in a million years did I think I would hear it the very next year in Bio 348 by my lecturer who taught me Bio 348 when they put on the slide supply and demand. And I was like, oh my gosh, I know. I didn't go and apologize to that environmental lecturer because they ruined my life for a semester. Economics was the death of me. So if you're ever wondering why I'm so mad, it's because of economics. No, I'm joking. Okay, so when you say supply and demand, we start now working on a process where photosynthesis has got needs, it's got demands, and in order for those demands to be met, there needs to be a supply. So if our supply increases, our demand for it decreases, and we have that healthy equilibrium in the middle. And that's what you need to bear in mind, that when photosynthesis is occurring, the plant's going to have demands for it. It's going to have energy. So we want photosynthesis to occur. It's nighttime. It's not helping. We demand energy. We demand energy daytime. All of a sudden, we have a nice rich supply of energy, and our demand for it decreases. We have a nice equilibrium in the middle here. So, but then, let's say you have ideal conditions. Ideal conditions, absolutely brilliant conditions to a T. It's beautiful, it's stunning. I can't explain to you how perfect it is. Photosynthesis is occurring. We've got enough water, we've got enough light, Calvin-Benson cycles happening, we've got enough generation, it's metabolizing, it's perfect. All of a sudden, the plant needs to grow because it's photosynthesizing, it's creating glucose, it needs to grow, it needs to make more leaves so it can photosynthesize more because it's got enough of everything. You need other stuff to grow. You need other stuff like nitrogen. You need other stuff like phosphorus. Because remember, what's an ATP? Adenosine triphosphate. You need phosphate. You need zinc. You need calcium for those cell walls. You need sulfur. You need all of this stuff. So now all of a sudden, your supply of nutrients starts decreasing because your demand is increasing. Your demand for that is increasing, your supply is decreasing. So something is always going to be limiting. You have to, as a third year applied plant ecophysiologist, you have to be able to look at the plant and be able to go, what is its limiting resource? What is limiting this plant at this moment? Is it photosynthesis? Is it light? Is it water? Is it oxygen? What is it? Is it nutrients? You're going to have to understand what is its limiting resource. And from that, you will start understanding how stress is going to relate to photosynthesis at later stages. So remember, recap this, the metabolism of this stage from G3P to sucrose is very important, and then we get this regeneration where we need energy to reduce it back or regenerate it back from the G3P to this 5-carbon molecule. Okay, now remember that we're talking about supply and demand, and these processes, this process of photosynthesis is going to occur in the leaf, and the leaf is specifically designed to handle photosynthesis. The leaf's main job is to photosynthesize, and because its main job is to photosynthesize, it needs to be adapted and its structure needs to be there in optimal condition to photosynthesize. So we have this demand that is being placed on by the cells, the spongy mesophyll cells, this demand for carbon dioxide, this demand for water, this demand for light, and the supply for carbon dioxide, for water will be stomata, it will be the xylem, it will be the light. So you have to recognize what demands are being placed and where is the supply coming from. The demand that's being placed on this for photosynthesis is we need water, we need carbon dioxide, we need light, we need what else do we need? I think I said it all. And the supply, the stomata is going to supply the carbon dioxide by bringing it into the intercellular spaces. The xylem is going to provide the water. The sun's going to provide the light that we need. So supply and demand. Having a look at the stomata. Remember, the stomata works on water gradients, so water concentration. So when it is turgid, meaning it's full, it's plump, the pore is open, so it's filled to its capacity with water. And if it's flaccid, the water's lost, the water concentration's lost, it's closes, it's flaccid, it closes itself, and it doesn't open. So turgid, open stomata, flaccid, closed. And this becomes important because when you have a situation in a plant where you have low water, so also taking a step outside of this for a minute, just take a walk with me. This is by far one of the only modules where you have to connect the lectures. They're not in little boxes. They're not in photosynthesis box, nutrient box, abiotic stress box, water relations box, and you keep them separate. You have to show a level of understanding and connect the boxes. You have to. This is applied plant ecophysiology. So you've got to think now, all right, I've got high temperatures, I've got low water, water relation. It's going to decrease the water concentration in the stomata. It's going to cause the stomata to become flaccid. It's going to close the stomata, meaning I no longer have access to carbon dioxide, which means my internal oxygen levels are going to increase. If I'm a C3 plant, problem. If I'm a C4 plant, no problem. But photosynthesis is going to come to a halt because all of a sudden my demand is carbon dioxide for photosynthesis to occur, but my stomata is closed because there's not enough water. So you need to be able to connect. So when you are turgid at full water concentration, the stomata is open, flaccid, little water concentration, closed. Okay, remember what I said to you? The primary function of the leaves is photosynthesis, and in order for photosynthesis to occur, the structure of the leaf and the function of the leaf needs to be optimal. So the leaf anatomy as well as the orientation is important. So we've looked at the leaf anatomy. We see how the leaf anatomy has allowed for oxygen, carbon dioxide to come in, light to be absorbed, etc., etc. But we also need to consider the orientation. And no, I do not mean sexual orientation. I mean the way it is facing. Okay, whether it's facing the sun directly, whether it's at an angle, whether it's at a side, whether it's concave, convex, etc. All of that is going to affect the absorption of the light. The chloroplasts are designed to assess the situation of light and either increase in number or decrease in number depending on what is perfect for that region, which we'll get to a little bit later. Remember, the photosynthetic responses are going to differ under different growing conditions, under different light conditions, under different water conditions, under different growing conditions, environmental conditions. The photosynthesis is going to change. So I'm going to show you later in the slides the difference between plants growing in same plant, same species, growing in different conditions and how it changes because of the different environments. So photosynthesis adapts itself to the environment that it is undergoing. Remember, always remember that photosynthesis is dependent and is limited by those demands. What it is demanding. It's demanding light, carbon dioxide, etc. So photosynthesis will be limited depending on how much access it has to what it demands. Also, plants can't just get up and move. If I'm hot, I'll go into a room with an aircon, switch the aircon on. If I'm cold, I'll get up and switch the aircon off and put a jersey on and migrate to the Bahamas. Plants cannot up and walk away. You know, plants are stuck. Where they are is where they are. And that's very unique. It's unique because they are sessile. And because they are sessile, they have to be able to adapt. Because the environment anywhere in the world, even if we didn't have climate change, even if we didn't have global warming or whatever the case may be, it's never going to be constant. It's always going to be fluctuating to a certain degree. And as long as it's within a certain degree, the plants can adapt. And their photosynthesis can adapt. And their photosynthesis can change in order to handle the conditions around it. However, there's limits to that. And that's what we need to understand. And the limits start coming into the fact that now we've got climate change, the thresholds are being broken and being breached. Can the plant still adapt its photosynthesis to handle the different growing conditions now? And so on and so on. Okay, so I am going to upload multiple of these because I can't hear my voice for an hour. I can't imagine how you can. So they're going to be in sections, also because it makes it easier for you to download or watch on YouTube or Moodle and easier to upload, etc., etc. So I'm going to stop here for now. I will